Hello, my dear students. Are you doing fine right now? I hope you are. We are now on our third day of our lesson, and we will be discussing about genetic engineering. Are you now ready? Let's watch a video first.
Using genetic engineering, scientists can create cells that produce proteins they wouldn't ordinarily produce. For example, circular pieces of DNA, called plasmids, found inside bacterial cells, can be engineered to make such proteins as human insulin by inserting the gene that codes for that protein. Special enzymes called restriction enzymes, which recognize, bind to, and cut specific DNA sequences, are used to open up a plasmid in preparation for the insertion of a gene. Some restriction enzymes leave the DNA with uneven ends, also known as sticky ends, shown here. If the gene we wish to insert and the plasmid are both cut with the same enzyme, these sticky ends will be complementary, meaning they will readily bond with one another. In a follow-up reaction, gene fragments, cut plasmids, and an enzyme that connects DNA fragments are mixed together. This so-called ligase enzyme attaches the sticky ends in the solution to produce circular plasmids that now carry the gene that codes for the desired protein. Note that the efficiency of recombinant techniques is not 100% and may lead to plasmids with no inserts or multiple inserts.
What was the video all about? Right. The video is all about genetic engineering. Based on the video, cells were able to produce proteins that they don't usually produce through the use of genetic engineering such as human insulin. Restriction enzymes are used to open up a plasmid in preparation for the insertion of a gene. If the gene you wish to insert and the plasmid are both cut with the same enzyme, their sticky ends will readily bond with one another. The ligase enzyme attaches the sticky ends in the solution, producing circular plasmids carrying the gene that code the desired protein. So, what's genetic engineering, by the way? Understanding the gene has led to the remarkable development of methods for changing a cell's DNA. A modern biotechnology called genetic engineering produces transgenic or GM crops of organisms. Scientists have developed methods to move genes from one species into another. Genetic engineering has already been applied to bacteria, plants, and animals. These organisms are engineered to be of use to humans. Traditionally, humans have manipulated genomes indirectly by controlling breeding and selecting offspring with desired traits. Genetic engineering involves the direct manipulation of one or more genes. Most often, a gene from another species is added to an organism's genome to give it a desired phenotype. Genetic engineering is a term that was first introduced into our language in the 1970s to describe the emerging field of recombinant DNA technology and some of the things that were going on. In genetic engineering, you are taking pieces of DNA and combining them with other pieces of DNA. This doesn't really happen in nature, but it is something that you engineer in your own laboratory and test through. So here are the steps involved in genetic engineering. These steps are already introduced and discussed in the previous video. So first, a plasmid or a ring of DNA is isolated from a bacterium. An enzyme cuts the DNA at the specific sites. Meanwhile, a gene for protein taken from another cell is cut also with the same enzyme. So the gene taken from another cell is inserted into the plasmid where it fits exactly. So this is now the recommended DNA. The recombinant plasmid is inserted back into the bacterium. So the new gene directs the bacterium to make a new protein product such as interferon. When the bacterium divides and replicates, it copies itself and the recombinant DNA. That would be all for today, and thank you for listening.